All right, so welcome everyone to the very final, at least in our invited faculty part of the Maryland Robotics Center's seminar series for fall 2022. Actually, for the whole year. This is the last one for our invited faculty. And so I'm very excited because today we have Professor Webster-Wood, who is an assistant professor of mechanical engineering and courtesy biomedical engineering at Carnegie Mellon University. And so Professor Webster-Wood got her BS, MS, and PhD from Case Western Reserve University. She was an NSF Graduate Research Fellow winner, as well as a Gann Fellow. She also was the recipient of a Ruth L. Kirstein NRSA postdoctoral fellowship. These are all like major things for the students in here, kind of like, wait, what are those? And so she's going to be talking today about her work with her Biohybrid and Organic Robotics group at Carnegie Mellon. So let's give her a round of applause. Welcome.
And thank you to the whole MRC for having me. Can you all hear me okay? Is that a good position? Okay, awesome. So it's my pleasure to be here for this seminar and to get to meet so many of you and see your really awesome labs. I've been handling a lot of demos that I assume were made by many of you, and they're all very cool. So major kudos. But today I really wanted to share some of the work that my lab has been doing, specifically looking at bioinspired and biohybrid approaches towards creating autonomous biodegradable robots. That's really where our long-term focus is as a lab. So hopefully this is interesting and exciting, and I keep you all awake in this afternoon seminar slot. But if it gets too boring, just like raise your hand and be like, Vickie, this is boring. We'll change topics or something. Who knows? It's an adventure, and we've got surprise animations. So, of course, at this stage of my career, even when Ryan says that I'm doing the work, these are the people who are actually doing the work. This is my fantastic crew of graduate and undergraduate researchers. And on the slides that I'm sharing today, this is really the result of their hard work in the lab and through the pandemic. Okay, so let's dive right in. So when we look at the world around us, we see that animals are capable of tremendous feats of agility, right? They demonstrate this level of strength and ingenuity, creativity, and learning that we really want in our robots, right? Even relatively simple organisms are capable of learning and solving complex problems. And so as an engineer, I look at these examples and I say, what are the fundamental principles that allow these animals to achieve these great feats of performance, and how do I capture these in my robots? Right. So towards capturing these capabilities, my lab is focused on bioinspired and biohybrid robotics. These are kind of two parallel research thrusts that we pursue. And ultimately, we want to be able to learn fundamental principles of actuation and control by first studying and modeling biological organisms, and then look at how we can harness materials directly from our natural world in robotic systems. So we look at the renewable biocompatible materials that allow animals to achieve the capabilities that we observe. We then try to actually build adaptable living actuators out of muscle tissue, as well as understand how muscle achieves the performance that we see kind of good enough across the board in actuation, and then capture that in synthetic approaches. And we've even started looking at how we can create energy-efficient living controllers through the use of neurons in culture. And ultimately, we want to bring this all together to build soft bioinspired robots. And my long-term career goal, if everything works okay, or probably works, best, who knows, is to create completely biodegradable autonomous robots. And so this is kind of the pipeline that has driven all of our research. For today's talk, I sat down and I was like, oh man, what am I going to share? There's all this cool stuff that the folks in my lab are doing. How do you pick, right? And as a PI, you don't want to have favorites, right? Because I love them all equally. But I decided to kind of break the topic down into three themes that have been the primary focus of my lab since we started at CMU. And so I'm going to start by talking about some of the work that we do in more traditional materials in the synthetic robotics space, where we're really interested in robots as models for biology. right? So these aren't robots that we want to get out in the real world doing a specific task, but rather, can we use engineering approaches to build controllable models to allow us to test hypotheses from neuroscience? And then I want to get into some of the biohybrid work that we're doing. And in particular, we're really interested in the biomaterials that will allow us to build these biodegradable robots and fabrication approaches for those. And if I have time, I'll wrap up with talking a little bit about our work in the controls space. So there's a lot going on, so let's go and dive right in. So in my lab, we see robots as not only functional systems for applications, but also as research tools, right? And for them to be research tools, we need platforms that mimic the principles that we see in nature to the best of our ability with the materials and control approaches we have at hand. And so we use our robots to address fundamental questions from neuromechanics. In particular, we're interested in how do animals generate multifunctional adaptable behavior, right? You put an animal in an unknown environment, and it's going to typically succeed. It's going to find food, it's going to protect itself, it's going to find shelter, right? In environments it's never seen before. And these are things that we need robots to be able to do. So to address this question, we are studying actual animal systems. But if any of you have ever done any biology-adjacent research, you know animals never do what you want them to do, right? They're very complex. And when we think about mammals, we've got billions of neurons and probably hundreds of little muscles, horribly complex system. So to answer these questions, a common strategy is to use a tractable animal model, right? This is a kind of simpler, and my neuroscience friends would hate me for using that word, a little bit simpler in a relative sense organism that demonstrates adaptable behavior. For us, that is this crazy cool critter. This is the sea slug Aplysia californica. They grow from about yay big as little tiny juveniles up to, I've seen them the size of house cats. So we can use them to study multifunctional behavior and control across dynamic scales. So little background. For those of you who don't know sea slugs, right? You know, I know you're all sea slugs experts, but just in case anyone in the audience. These are Aplysia californica. They're a robust marine sea slug. They have a relatively small nervous system. In particular, we study the feeding apparatus that you see in this lower video here. This is a biting behavior. It's kind of like the mouth from Alien. It's coming out, grabbing seaweed. The whole thing is controlled by only 4,000 approximately neurons, right? As a result, there have been decades of neuroscience research on this organism. So there's a lot of literature that we can pull from in terms of understanding the mechanics and understanding the controls. As a mechanical engineer by training, these are also fascinating to me because they have a completely soft-bodied musculature, right? All of it is muscle attached to muscle, essentially. And it demonstrates multifunctional behavior. In particular, there's 3 behaviors that we're interested in. Biting, which is what you're seeing in the video. That's an attempt to grasp seaweed. Swallowing, where it successfully grasps seaweed and it pulls it in. And rejection, where it detects that the seaweed's not actually edible and spits it back out. Right? And you can think of this as a model for adaptable grasping, right? So it could be kind of more broadly expandable to other grasping robot systems. So this is the muscular system, that doesn't work, that we're interested in studying. It's called the buccal mass. It's the feeding apparatus. It has kind of 2 levels of control. One you can think of as a motor control ganglia, and the other, the cerebral ganglia. That's kind of the higher level brain, the closest thing that the slug has to a brain. So as an engineer, in my group, we kind of break this problem into 2 parts, right? On one hand, we have this multifunctional neural controller, and on the other, we have this soft musculature that provides morphological computation, which is the idea where the mechanics of the structure actually offload some of the computation from the controller. So in order to develop these control models, because this is a model for biology, we don't just want a controller that achieves the task. We want a controller that lets us understand how the brain works. Right? So our goal here was to predict and control multifunctional behavior in real time for our robot, using principles from the biology. So to do this, we need our tractable animal model, sea slug. We need a computationally efficient model framework. And our solution was to create a hybrid Boolean continuous model of the feeding behavior by combining Boolean approximations of neurons with a biomechanical model of the musculature. So what does this look like? Well, in order to do this, we really have to work really closely with biology collaborators. So working with these biologists, we're able to identify key neurons and muscles from the behaviors that we're interested in and the neural recordings. So this is what this kind of looks like when you take recordings from the animal. And we can see that specific neurons are responsible for specific aspects of the behavior. For example, here we start with pinching. That's like closing the front on the seaweed so it doesn't get pushed out. That little internal alien mouth gets pushed forward in retraction, closes on the seaweed, and pulls it back so that you get a swallow. And that behavior is kind of repeated. And we can identify specific neurons consistently throughout this behavior. Right? Well, this would be incredibly complex to model, this signal that we're seeing here, right? So we take a approach that we call demand-driven complexity to try to break this down into a more tractable problem, right? So fundamentally, at the most abstract level, these neurons are either on or off, right? And so we can translate each of these into a Boolean burst, right? Where 0 represents quiescence, the neuron is quiet, and 1 represents firing. And if we think of each of these as kind of a Boolean node, you can build essentially a finite automata, right, or a state machine of these logic statements. Right, so we build this network up in layers from the muscles. So we've got a motor neuron layer. It's controlled by some inner neurons that kind of coordinate activity of motor units, and that's controlled by higher level decision-making units, right, that take in external sensory cues. It controls the musculature, and then you get sensory feedback. Right? And so with these 20 neurons out of that 4,000, right, we were interested in seeing, well, can we connect this to a biomechanical model and reproduce the behavior that we see in the animal? Right? What level of detail do you need for your model to capture animal behavior? So we connected it to a really simple biomechanical model. This is just kind of a cartoon sketch. Whenever you're working with biologists, it's really helpful to make sure you have a shared language, right? So we want not just to use engineering diagrams, not just to use pictures of animals, right? But what's the happy medium? Ta-da. So this is a little slug. During the behavior, it starts at rest, protracts forward, closes on the seaweed, retracts back. Right? And we've got equations of motion treating each of these as point masses. Right? When we connect this to our control model, right, even with this really relatively simple model, you can actually start generating these complex behaviors that you see in the animal. Right? So if we mimic the stimuli that would trigger the animal to bite, right? So if there's chemical smell of seaweed in the environment and it kind of feels seaweed brushing against its face, the slugs will start biting, right? And we can reproduce that in our model. If you then add mechanical stimuli, surprise animations, I told you. We'd be surprised by them together. Key features of biting. If you add mechanical stimuli that would mimic successfully having grabbed that seaweed, right? So now you've got pressure in your grasper, you get a different behavior. You start generating force on the seaweed, right? And we see the same thing in animal experiments. And here we've got these 3 positive stimuli, and we see strong force development while the retractor is being pulled in order to swallow. And then we can also reproduce rejection when there's just mechanical stimuli but no chemical stimuli, right? This would be like if you tried to feed the sea slug a plastic tube, right? And now we get this strong outwards force while the grasper's pushing forwards, so it's spitting the object out. And our biology friends say that these look very similar to the force traces that they see in the animals. Excitingly, even though we didn't program this in at all, an emergent property of this network, when you use the connectivity and the musculature that you see in the animal, is that you get emergent behaviors, right? So here, the network is capable of behavioral switching with no additional tuning, right? So we can change the sensory state, in this case from a bite-like state to a swallow-like state, and the behavior transitions smoothly. Right? So this is all super cool, but of course the animal is not being exposed to just this little simple simulated piece of seaweed, right? Seaweed has different strengths, it has different sizes, what happens to the model when we expose it to those types of environments. Well, when we expose it to weak seaweed, so here we've added a force threshold so the seaweed can break in the simulation. Now you get these bite-like patterns. So you've got the grasper motion, it protracts, it retracts, but you see that little seaweed break there on the force seaweed. If we then strengthen that threshold and allow higher forces on our simulated seaweed, now we get these more swallow-like behaviors, right? So we've got this, again, high force developed. Okay, so that's all pretty cool, but that was a little bit of a neuroscience talk, right? You're like, man, I thought this person was a roboticist. Ryan, what are you doing, right? Well, so we see these emergent properties from the control system, right, with relatively simple control system. And then the question is, well, how much of that do you actually need for the behavior, right? How much of that was that control model and how much of that was the biomechanical model, right? And when we study these animals, right, I'm a basic scientist, so I just get like crazy excited about how the world works. We see these really weird shape changes in the feeding apparatus during feeding behavior, right? And so the grasper actually changes shape, and that changes the mechanical advantage of the muscles, right? So activating one muscle changes shape, changes the mechanical advantage of another muscle, and now you've got completely different equations of motion, right? So we were really interested in this question of like, what matters more? It's biology, so the answer is always yes, right? There is no, it's that or that. And so we wanted to look at what happens if we add complexity to our model, right? So my PhD student, Ashley Liao, and Ravesh Sukhmandan, who at the time was a master's student and is now continuing for his PhD, developed this super modular slug in a computer. And so they can plug in these different biomechanical models, different control models, and compare their behavior to animals. And what they see is that with the original model that I just showed, as we saw, you can get all three interesting behaviors. This model just translated, right? But in the animal, we see this rotation too, right? So as you start adding those degrees of freedom, you see you still get these behaviors, but your dynamics start changing, right? And then as you start adding more biologically plausible muscle models, again, the dynamics of the behavior change, right? And this is without changing the controller at all, right? So something is going on in the mechanics that's important, but what about the mechanics is affecting this the most, and how does that affect feeding, right? Well, these questions start becoming very difficult to answer in simulation, right? For those of you who do numerical computations, you know that those are very often long runtime experiments, right, to do a big simulation. This is completely continuum model, right? We're abstracting things as springs because we want it to run fast, but if we actually modeled this in FEA, it'd be crazy, right? And no matter what we did, right now we would have a simulation reality gap, right? So we've got these hypotheses about which components of the mechanics are most critical to the timing of feeding behavior that we can't really address in silico at kind of real-time speeds. And this is where robots come in, robots for biology, right? So one way, potentially, to overcome the simulation reality gap is to build in roboto, instead of in silico, models of your organism. And so my group does a lot of this. We look at the specific, I don't know why I keep pointing with a laser pointer at a screen. We look at the specific muscles that we think are critical to the mechanics, and we reproduce them with robotic actuators. So this is a fully pneumatic version of the robot that we've built. We include key muscles. These are intrinsic muscles. You can find them in every single animal. For linear intrinsic muscles or for these toroidal ones, these are just your standard McKibben style actuator. You've got a braided mesh that controls your contraction direction. For the I2 in the animal, this is a really broad, flat muscle. And we think that the mechanics, the stretchiness of that muscle is important for the robot. So we've done this with a take on the flat PAM concept, where we've then created these big arrays of flat PAMs so that we can get fiber orientations to match the animal. This is just created through kind of a series casting approach so that we can use this polyester film to provide the contraction orientation. Then we combine this with functional approximations of the parts of the anatomy that we don't know all the details for, right? So we're as biomimetic as we can, where we can be, where we can't be, we do functional analogs, right? So this is the grasping apparatus. There's a ton of little muscles in there that we're still working on characterizing. So for now, we've just got openers and closers. And thus, SLUGBOT is born. This is our slug-like uniaxial grasper robot. As a grad student, you should practice your acronym game, right? They have a lot of fun with it. They come up with crazy acronyms all the time. And so we can then attach this to our neural controller and get slug feeding. Slugs are slow, right? But we were able to reproduce, again, these feeding behaviors in response to these different input signals, both for biting and swallowing. And perhaps most excitingly, our V1 version of SLUGBOT does a pretty darn good job of actually reproducing kinematics that we see in the animal, right? So this here, in the blue trace, we've got our robot data. In the gray trace, our animal data. And we're getting fairly decent agreement. Now, we do see that there are some differences in the dynamics here. And this then kind of creates this iterative loop between us and the biologists, right? So now we need to go back to the biologists and say, hey, our mechanics weren't right for this part of the cycle. Can we get more data on how that muscle actuates, right? Maybe we've got the wrong mechanics for that actuator relative to the scale of the robot we're building. And so we can iteratively keep refining this until it matches the animal. Okay. So this has all been bioinspired robots. We're using traditional synthetic approaches, elastomers, plastics, metals, all of that, right? But there are some limitations to these types of approaches when we're thinking about biomimetics, biodegradability, biocompatibility, right? The synthetic actuators that exist in robotics, despite the fact that what all of us actuator people will tell you, they don't really yet reflect biological muscle, right? They're usually good in one area, right? Maybe they've got a really great strain ratio or a really great power density. But muscle is kind of good enough across the board, right? Like it's a darn good actuator. Doesn't excel in anything, but really good at what we need it to do. Synthetic actuators also currently lack adaptability in response to training, right? We can get stronger. Synthetic actuators typically can't. And many of them lack self-healing. I say many because there are now some really cool polymers that self-heal. Super awesome. And then our controller was very simple, right? So our neuron dynamics don't really match actual neurons. We've only included 12 to 20 depending on our control model, and they can't learn yet. So these are some limitations. We are continuing to work on addressing them in synthetic approaches. We're developing better control models, better actuators. But I really want to kind of turn away from this idea of bioinspired robotics for the remainder of my talk and look at this idea of biohybrid robotics, right? We want a muscle that's like, an actuator that's like muscle, right? Why not just use muscle, right? And this is motivated for me by a couple of factors. And it really starts by looking at what goes into a robot. Right? Well, when we build robots, you know, we've got some sort of chassis, probably need some actuators, sensors, a controller, right, electronics. And each of these is going to require metals and plastics and rubbers, elastomers, wire, magnets, and then when we look at electronics, things like copper, lithium, platinum, and unfortunately, many of these materials are hazardous, right? You know, obviously we don't want lead and chromium in our electronics, but the reality is that recent surveys show that most of our consumer electronics have levels of lead and cadmium and chromium above regulation standard, right? Unfortunately, many of these materials are also non-renewable, right? They have limited availability in natural reservoirs. So recent studies have tried to predict the long-term production peaks of many of these components. And we see production peaks even within the next century, right? Copper might be as soon as 2072, right? That's a big deal. They're difficult to recycle, right? Many of you have probably heard about the challenges of lithium recycling and how that's such a big problem, especially as e-vehicles become more prevalent. And they're continuously facing increasing demand, right? Now, of course, we as roboticists are not the main driver of this demand. It's consumer electronics, phones, computers, all of that. But I think we need to be aware of this, right? And as we're increasingly taking these minerals from mines around the world, we're also seeing decreasing ore grades, right? This is going to affect electronics of the next century, right? So we want to think about this when we're thinking about maybe not robots of 10 years from now, but robots of 50 years from now. How do you as trainees lay the foundation now to address these challenges? Right? Many of these materials are also often hazardous or environmentally damaging to mine. And local disasters or tragedies can have tremendous effect on global supply chain. Right? We saw this with the pandemic. Right? And unfortunately, the reality is that these events are not going to stop. Right? We're going to potentially continue to see increasing weather events, increasing health events, all of these sorts of things. Right? So it's a little bit of a downer for the middle of the afternoon, right? But someone has to do the basic science now to allow us to have solutions for when these things really come to a head, right? So after you all build your robots and you graduate and you go out into the real world beyond the lab, what happens to your robots? Well, in my PhD lab, we had shelves and shelves and shelves of ancient robots, right? The robots may break, they may leak, right? They may be used and discarded. Sometimes, you'll cannibalize them and keep recycling them because you need that motor for this year's robot, right? But ultimately, if we've done our job correctly, our robots should become obsolete, right? Because we want to keep advancing robotics. So all of this e-waste, all of this material waste has to go somewhere. As someone interested in bioinspired and biohybrid robotics, I again kind of look back to nature when faced with these types of challenges, right? And unlike robots, biological systems, animals, plants, all of that, right, are composed of dynamic, responsive materials, right? The structure of our tissues is guided by biodegradable proteins. Our actuators are adaptable and self-healing. And you can even think of the brain as a dynamic, intelligent material. Right? And this brings me kind of to the second half of our work, right? Which is how can we leverage the desirable properties of these living materials directly in sustainable, renewable, biodegradable robots? So this problem really starts with materials. In my lab, we're interested in growing muscle-like actuators or muscle-based actuators. And so we really want active, adaptable robotic materials that are biocompatible, renewable, and biodegradable that we can use with living muscle tissue or with living neurons. I think this is particularly important for applications in things like biointerfacing and medicine or environmental deployment, where, we want to be able to deploy all these robots without harming the environment that they're in. So one material that we think shows great promise for biohybrid robotics is electrocompacted and aligned collagen. So this is collagen like is in our skin or in our tendons. It's an extracellular matrix protein. And we can actually form pretty robust materials from this protein through electrocompaction. What we do is we introduce a viscous molecular solution of collagen between two electrodes. We apply a current across those electrodes, and the molecules become charged. And the local forces on the molecules compact them along the isoelectric point. So in this way, we can produce really robust threads or sheets of collagen. And the threads can be chemically cross-linked and actually be strong enough even to use as sutures. What's really interesting about this is because it's an extracellular matrix protein, cells love attaching to it. And if the collagen molecules are aligned and you grow cells on them, the cells become aligned. Right? And so we can use this as kind of the platform for robotic structures. So during my PhD, we showed that you could fabricate kind of arrays of these compacted collagen sheets and seed muscle cells on them. And then after several days, when the muscle cells kind of grew up and matured a little bit, you could electrically stimulate these and get them to move. Very slowly. A lot of my robots are slow. So this kind of shadowy thing is the collagen sheet. It's seeded with cardiomyocytes or skeletal muscle cells on the bottom side. And then over time, it kind of scooches forward as you electrically stimulate it. So this was a great proof of concept, but there's some problems. We saw that these robots really had limited deflection of the legs, and they had really low muscle force that was being generated. Right? And so to address the challenge of limited deflection, maybe this is a mechanics problem, right? Maybe there was just too much collagen. It was too stiff. The cells couldn't bend it, right? So when I started at CMU, we came back to this problem. And my PhD student, Wen-Hun Sun, along with master student Jason Polovich, really looked at how can we, from a manufacturing standpoint, tune the mechanical properties of these collagen threads towards properties that are more amenable to muscle-based robots. So we've got a lot of fabrication parameters that we can adjust during manufacturing. For example, we can vary how long we apply a current, we can vary what voltage we apply, or vary the starting concentration of the collagen. And so they wanted to know, how does this affect the geometry and the material properties of the resulting threads? So they ran a lot of experiments. I'm not going to go through all of them, but if you're interested, through the citation up there. And what they found was that we really could tune these threads. So for example, if you had a longer IF duration or a higher collagen concentration, you could get higher tensile strength. If you fabricated these fibers with a lower voltage or a lower duration, you got kind of smaller threads with weaker mechanical properties. And by kind of introducing cross effects between all of these different manufacturing knobs, we could really tune the mechanics of threads across quite a wide range. So folks who aren't in bioengineering, up here, this is more like tendon. Down here, not quite as soft as muscle, but closer to muscle. So this was great. This gave us a lot of hope that we could actually start to design robots using these materials and then be able to actually reliably produce them. So Wen actually also developed a deep neural network that you could say, hey, I need these mechanical properties, and it would tell you, use these manufacturing parameters, which is hopefully going to be very helpful. So we think this will help address kind of that limited deflection that we're seeing. But what about the low muscle force? And it turns out this is kind of a known problem in tissue engineering. Right? So in early biohybrid robots, everything was done with 2D culture. Right? So you grow these cells on a flat surface, and what you end up with is contractile myotubes, which are like kind of muscle precursors. And they contract, but they're really immature. It's kind of like embryonic muscle. In contrast, if you grow cells in 3D and try to reproduce the environment that they're actually in natively in living organisms, you get much more mature muscle with better performance. And this is a trend that we've seen play out in the field of biohybrid robotics. In the early days, all of the robots were these kind of cantilever-based systems where you have muscle cells on one side of the structure and then contraction of the muscle cells. It's a bilayer cantilever. It's going to bend, right? And since then, the field has really moved forward towards 3D muscle tissue. And 3D muscle tissue can be fabricated typically by casting or more recently 3D printing. And there have been some really exciting biohybrid robots produced with these techniques. But there's still a challenge, and it's the challenge that you're never going to see in the final papers, right? Because all of these biohybrid robots right now are typically being fabricated with the ever-present grad student with tweezers method of fabrication. And you've got a soft muscle connected to a rigid robot. What's going to happen? It's going to tear. right? We're introducing these stress concentrations in the muscle. So the field typically has fairly high failure rates for the number of robots that can actually be successfully fabricated in any batch. And there's a critical need, I think, for better manufacturing techniques that will allow you to connect these biohybrid actuators to a wider range of robots, right? How am I going to convince you all that you should put muscle on your robots, right? Well, I can't tell you that 1 in 10 is going to fail, right? You'll never switch away from McKibbins. So this is an area that we've started trying to combine our prior experience with the biomaterials, with our experience in manufacturing, in order to create 3D muscle tissues with tendon-like ends. So we've been working a lot with the freeform reversible embedding of suspended hydrogels printing method. This was developed by Dr. Adam Feinberg, who co-advises Wenhun Sun. And for those of you who maybe aren't familiar with this approach, the idea here is that you have a support bath that you embed hydrogel bioinks into, and then the support bath holds the shape while they cure, and you can cross-link them and all of this. And then you can release them. We use a gelatin support bath, so just with temperature release. And this approach is great for bioinks because it's compatible with a lot of our standard extracellular matrix hydrogels. But as with any hydrogel manufacturing approach, the resulting printed structures are fairly weak and soft, right? Just like our muscle tissue, right? So what we've been working on is combining this printing approach with our collagen biomaterials. And so we've developed a new fiber reinforcing printhead that allows these prefabricated collagen fibers to be embedded into the hydrogels during printing. We combine this with a Repl-Extruder syringe pump for extruding the bioink and a open source 3D printing platform to create a multi-material fabrication platform for long fiber embedding during fresh. So this is kind of the way that the extruder works. The fibers fed through a series of guiding tubes and then gently pushed out by these silicone wheels. And that allows us to take one print head, print the hydrogel, embed fibers, print more hydrogel, etc., repeat. And then following the same kind of fresh approach, we liquefy the support bath and retrieve our parts. Of course, this process is kind of like trying to push a rope into jello, right? So there's a lot of challenges with matching up your material properties to get successful extrusion. If the thread is too weak or the gel is too stiff, the thread actually buckles during the extrusion, right? So we've developed both analytical and computational models of this buckling process so that we can kind of provide guidelines on what fibers are actually printable with this approach. And then we experimentally validated this, which is the checks and X's. And we get okay geometric fidelity of these embedded fibers, right? So here we've just used OCT imaging to 3D reconstruct the embedded fiber, and we get deviations that are sub-millimeter, which is okay for some applications, not okay for others. You know, choose the fabrication platform that works for the resolutions you need. We can improve the print fidelity by using closed-loop computer vision-based control of the printer. So we can detect buckling and change the extrusion rate in order to minimize kind of deviations from the desired print path. You can also use a number of techniques from tissue engineering to improve the mechanics of these composites. For example, chemical cross-linking can be used to improve the bonding between the fiber and gel, if that's important for your application. And as we anticipated, using this type of structural reinforcement improves the mechanical properties of the composites. So you can embed these fibers and greatly increase the ultimate tensile strength of your composites. And as you might expect, this also kind of translates to bending strength. Okay. We've tested this printing approach with a number of different hydrogels and thread types that may be of interest to the soft robotics community. And to biohybrid robotics in particular. So we've used alginate hydrogels and collagen hydrogels. Our collaborators also printed with PDMS. And then we can embed fibers that are, single ply, triple ply, nylon, polyester, silk, really little metal wires. I think it's on here. Yeah, you can see this very strange silver nanoflake alginate composite that Wen was like, I'll just try that. And it's conductive. It's pretty cool. You can print one fiber at a time or multiple fibers in both kind of 2D and kind of layered 2D, so like 2.5D, as well as full kind of 3D embedding of these fibers. So this platform now, what we're doing is translating this towards biohybrid robotics by combining these printed tendon-like ends with actual printed muscle, which is too early for prime time, so you just get this teaser video. Okay, I think I have a tiny bit of time left, so I will wrap up with a little bit of a look at the work that we're doing in the controls space. So we're really interested in both taking principles of control from studying living neurons and translating those to in silico control, as well as working towards growing networks of living neurons as robotic controllers. And this was really motivated by some preliminary work that I had done that showed that when we took these muscle tissues from sea slugs for use on robots, we got much better performance of the muscle if we stimulated via inducing natural motor patterns in the neurons versus if we tried to electrically stimulate the muscle. Right. And this is something that the functional electrical stimulation community has seen kind of consistently, right? If you stimulate with the really old electrodes that went straight into the muscle, you got fatigue really quickly, but nerve cuff electrodes reduced fatigue, right? So it's the same concept. And so we're trying to translate this into biohybrid controllers. In order to build biohybrid controllers, we really needed to start by understanding neural growth, right? What are the types of patterns that you see in neural growth? How can you constrain that and kind of induce neurons to grow the way you want them to? And how can you build design tools that will predict neural growth, right? So we've created a data set of neuron growth in vitro in order to identify quantitative descriptions of different growth stages so we can compare to design tools that predict neural growth in silico. And in this work, we've looked at both kind of common morphometrics for the structure of neurons, the number of tips, the total length of all the projections, tortuosity, and we find that total neurite length and neuron degree vary significantly between these growth stages. And so now this gives us metrics to go look at how our design tools work in actually capturing neural growth. We've introduced some new morphometrics based on the change point test, which you may know from studying animal behavior or robot behavior. Right? It's the tendency of an animal to undergo goal-directed motion. And again, we find that we can kind of significantly identify which stage of growth the neuron is in based on these morphometrics. Okay. In parallel with our experimental work, we are building models of neural growth. This is work that's being primarily done by Quanren Qian, who is co-advised by myself and Dr. Jessica Zhang. And he's developed a phase field model of neural growth using IgA collocation in order to actually kind of try to reproduce how these neurons are growing in simulation. And from this, then, we hope to be able to actually pattern networks in the future. And then I guess just one last thing to wrap up. I'm at CMU. I had to learn how to do machine learning. I think that comes with the job description. And so what we've also been doing is then taking these principles that we've learned from looking at the neurons growing in the dish and bringing them back into the computer. Right? And so this led to the development of a growth initialized neural network approach. And the idea here is that standard neural networks typically have these fully connected structures, right? Or they're like randomly set to sparse network architectures. But of course, the brain is very sparse, right? Very, very sparse. And so we wanted to look at whether growing these neural architectures for more traditional ANNs could improve their performance, either in terms of improving accuracy or in terms of reducing computational cost. So my PhD student, Kevin Dye, developed kind of two approaches to grow neural network architectures. First being stochastic grins, where you just kind of randomly set axon growth rates and directions. And the second being an adaptive method, where the growth direction varies depending on the simulated chemical cues in the environment. We tested this on the MNIST kind of benchmark data set for image classification, and interestingly found that growing these networks improved classification accuracy over the fully connected network. So we're continuing to work on this project, so we'll see where it goes from here. Hopefully we'll keep adding more biological mechanisms into this growth. But moving forward, where's this field going, right? I think there's a number of ongoing challenges in biohybrid and bioinspired robotics. Okay. You know, when we look at these materials, all of those muscle-powered robots that I showed you, they live in a sterile incubator, in petri dishes. They have to be handled in a sterile hood. If you expose them to room air, the bacteria gets in and just chows down, right? So we need to develop better packaging and life support mechanisms, right, where there's opportunities for contributions from additive manufacturing. And then we need to be able to scale that manufacturing up, right, so that these are useful techniques beyond the lab. In order to convince roboticists to use muscle actuators in their robot, we really need like the equivalent of a Dynamixel, right? You need to be able to tell me, hey, I need an actuator that can provide me with 1 Newton of force and 10% contraction strain. And I need to be able to say, okay, here's the build plan, right? And then with these controllers, this is really early days, right? There's a lot of work in this space and room for contributions, both from roboticists, from modeling, from neuroscientists. But ultimately, again, this is really kind of in this goal of creating autonomous biodegradable robots. So hopefully sometime in the next 50 years, we'll get there. And hopefully I've convinced some of you that you should join me. So just wrap up. Again, want to acknowledge the people who actually do the work and our collaborators, a bunch of collaborators at CMU as well as externally, as well as our funding agencies. And a little shameless self-plug, any master students or undergrads in the audience, we are recruiting this year. Deadline's December 15th. Got 2 positions open.
I know there's a lot of undergrads in here, so feel free to stay behind for those cases.
Cool, awesome. Well, and I'm happy to take any questions.
Let's do a round of applause while I pick up my microphone. All right. Turn this back on. Okay. I feel like mine is much louder than yours is.
I think mine's further away. I think mine's on.
Okay, it's on my belly button right now. I feel like it's still, I don't know. All right, so we got you a little Bioinspired gift.
Thank
You want to take a look? See if you can be a detective and figure out which animal it's been inspired by.
I love it. I love all your turtle stuff.
Yeah.
That's fantastic. Thank you.
So thank you so much for coming. All right, so for the people who've been watching online for the first time, I've been connected to your questions. I know there's been some frustrations the entire semester with me not getting your emails, but today is the first day that if you want to send questions, I will actually get them, supposedly. It might happen tomorrow, but I think there's a chance that I could get it today. In the meantime, any questions from anyone here about anything that you've seen today or you want to ask? Yeah.
Great talk, thank you.
Thank you.
So I was wondering, like the system you presented at the beginning, is it closed loop? And if it's not, do you have any thoughts on if closing the loop will help you improve the results?
Yes, I'm going to repeat your question for the people online. But the question is for the model of the sea slug, where we've got the nervous system and the biomechanics, is that closed loop? And how does closing the loop or not closing the loop change the results? Right? And that's a fascinating question because we actually see it in the neuroscience too. A lot of neuroscience research, they used to just take ganglia out of the animal and study it and say this is the way it works. And what our collaborators have found is that you get vastly different motor patterns if you do that versus if you have sensory feedback. So one of the things that we actually really made sure to do in that model was have sensory feedback pathways. They're very abstract in that model. Basically, we feed back just what is the position of that grasper within the head of the animal, and the model has access to like the grasper pressure, right? So, and part of that is because we don't know what those pathways are in the actual neural circuitry, right? So we kind of, as modelers, one of the nice things about being modelers is you get to take your best guess and say, it might be this. And then the biologists have to go find it in the actual animal. But so we did try to close the loop. I collaborate really closely with Roger Quinn's group at Case Western, and I've been working with one of his PhD students, Hyunjin Lee, to build like the next version of this controller. And he's actually looked at what happens to the motor patterns with and without that closed loop control. And you see that the timing of the behavior completely changes. Right? So I think closed loop control of these systems is really important. We need more information about like where are the sensors in the animal and what are those sensory feedback pathways to the inner neurons.
One comment, if I may. So I really like the degradable materials that you presented, but I just wanted to comment that sometimes it's also, especially for soft robots, it's the fabrication process also something that we should be concerned about.
You're absolutely right. So the comment was that the fabrication process for soft robots is a problem in and of itself, right? There's a lot of chemicals that we use in those, right? Yeah, and so I think one thing I didn't include in these slides, I've got some other slides where I look at all the possible plant and animal sourced materials that we could be looking at using. And if we think about alternatives to polymers, alternatives to elastomers, right? There are really interesting materials in nature that we could use as alternatives. The alginate that we're printing, we've been now working on printing more actuators with that. That's derived from seaweed. And basically all we add is water, right? So could potentially be a more bio-friendly fabrication process. And there's other options, right? So in nature, like you've got exoskeletons of insects. Those are much harder. Could we build with keratin? Maybe. Maybe we could build with chitin. I don't know. We got to try. But we need better manufacturing approaches for those too, because getting those sometimes, again, takes crazy chemicals to just like extract them from the tissue.
Great, thank you.
Yeah, great questions. Any other questions? Yeah.
So the work you're doing seems inspired naturally by the evolution that has taken place over hundreds of millions of years to get to something that works really well. Is the next step to then take the controllers that you guys have already built and then do synthetic evolution to try and get to more desirable traits in these biomechanical robots?
Absolutely. Yeah, evolution, I always tell my students, evolution is not optimization, right? Evolution is good enough-ization, right? So it's definitely not going to be the best solution to a problem, right? But it's a workable solution. So, and that's where this idea of general principles comes from, fundamental principles, right? Yeah, so once we have a system that works, can we use engineering optimization or genetic algorithm evolution to, I guess, as you say, evolve in silico to the next iteration? We're actually doing that from the sea slug robot. The SLUGBOT you saw, that was part of our neuroscience grant, right? Where we want that to match the animal. And we don't care if it's a good grasper, as long as it matches the animal. And then we got this other FRR award, actually, for exactly that idea of like, well, we think we now know how this works. Let's take that as the basis, evolve it, optimize it for agricultural grasping. Right? And I think there's a lot of potential across the board for thinking about research like that, right? Legs are great. What's the evolution, right?
Great. So maybe last question before we end. Are there any particularly unexpected applications of your research that maybe, like I'm going to a holiday party, to one of those ugly Christmas sweater parties tonight. Is there something I can talk about, you know, that people would just be like, wait, you're doing what with what?
Applications of the research.
Not to put you in the spot.
Yeah. Well,
So-
and as a basic scientist, the applications question, I'm always like, right? I don't want them to have to be good. I just want to know why they work. I guess crazy thing, Wen has been, like I said, printing some alginate actuators. He just, the last couple months, fed like 30 of them to sea slugs to make sure that they were safe.
You've been feeding your actuators to the
sea
slugs?
And then in his defense, he comes and he has a picture of, not to be gross, slug excrement with the pieces of seaweed excrement and the pieces of actuator excrement. And he goes, look, it's edible, to his PhD defense committee.
So clearly renewable and part of the new ecosystem.
Yeah.
That said, at least for those of you working on actuators, don't
Yeah, that's
don't
what we're
eat your
targeting.
actuators.
Underwater robots, marine applications, sensitive ecosystems like coral reefs, right? Where I don't want to release a swarm of robots, have them break down, and now I've killed a bunch of sea turtles, right? But if my robots are made of seaweed, not a problem.
That is, I think that's pretty cool. All right, so we're gonna stop there. So feel free to stay behind for those of you who would like to ask more questions directly. Otherwise, that is it, at least for this semester. So it's great having everyone who participated in these, and see you in the new year. Bye, everyone.